Hi, my name is Rongting Huang. I am a PhD candidate from the University of Hong Kong. Today, I would like to introduce a new developed tool, XClone, on detection of allele-specific copy number variations from the single-cell RNA sequencing data. As we all know, the copy number variations, including the copy gain, copy loss, and copy neutral loss of heterozygosity, have been widely shown associations in various cancers. Therefore, the accurate detection of the copy number variations is crucial for studying the genetic factors that contribute to the development and progression of the cancers. And now, single-cell RNA sequencing technologies provide a way to get a dull readout of the transcriptome and the genetic makeups. However, the single-cell RNA sequencing data sparsity result in the same property in both the read count and the allelic count. So the high technology sparsity makes it challenging to call the copy number variations at the single-cell resolution accurately. So we developed the XClone, which aims to detect the haplotype-aware copy number variations and try to reconstruct the tumor clonal substructure by integrating the expression levels and the allelic balance information. It is obvious to infer the DNA copy number events from the RNA sequencing because gene expression level can provide the depth information. For the copy gain, we expect more transcripts than the normal state. And for the copy loss, we expect less transcripts than the normal state. Generally, the copy number detection based on the expression profile compare the read depth with the reference cells to get the read depth ratio. And the read depth ratio higher than 1 means the expression increase and less than 1 means the expression decrease. However, just depend on the expression profile cannot help to distinguish which allele have the copy number variations. Actually, based on the genotype, the heterozygous SNP, we can get the B allele frequency information from the UMI counting, the allele count and the total count. And based on the allele count and total count, we can get the B allele frequency. And when we just see the expression level, for the normal expression level, we can see that the B allele frequency is around 0.5. But if the B allele frequency shift from 0.1, then there can be a copy gain, a copy loss, and can also have a very special situation, that is the copy neutral loss of heterozygosity. So the B allele frequency is helpful in calling the important allele-specific mutations called the copy neutral loss of heterozygosity in cancer. This kind of situation can be missed if just based on the expression profile. So combining the B allele frequency can enhance the copy number variation calling in both the loss of heterozygosity detection and can more accurately detect the copy gain and copy loss. There are 5 methods to the copy number variation calling related task in the single-cell RNA sequencing, and I benchmarked the methods on 3 datasets and got the results here. Through Casper and Honeyberger count the B allele frequency in, but their result is not so good, and the InverseInV and copycat is good. Well, they just use the expression profile. Non-byte is good and can detect the loss of heterozygosity and can detect the copy gain and copy loss, and its accuracy is good, but non-byte can fail in some specific situation. To get more accurate copy number variation detection results, we developed the XClone. The XClone workflow begins with the single-cell RNA sequencing raw files to generate the allele-specific read count matrix and the gene expression read count matrix. The features by their genomic position and the cells by assigned cell barcode order. We tried the negative binomial modeling in the read counts and the beta binomial modeling in the 3 steps phase the B allele frequency. Then we integrate the information into an HMM framework for the copy number variation state calling. XClone performs the B allele frequency model and the red depth ratio model independently and combines them to call the final copy number variation states. Here shows the combined strategy of the expression and the allele-specific profile in XClone. We combine the copy gain, copy neutral, copy loss in the RDR module and the allele A bias, allele balance, and the allele B bias states in the BAF module. And then XClone can finally detect these general copy number variation states. Here I need to highlight the methodology of phasing and smoothing in the B allele frequency model. We utilize a 3-level allele phasing approach to enhance the signal-to-noise ratio when analyzing the allele information, and the phasing helps to get a more accurate haplotype allele frequency from the SNP level to the gene level and then to the bin level. We can intuitively feel the function of the phasing and smoothing through the visualization of B allele frequency at different stages. Here I show one example from a glioma sample dataset, and the panel A here shows the B allele frequency after the reference phasing. It is at the gene scale. And the B shows the B allele frequency after the local phasing. It is at the bin scale. And the C shows the B allele frequency after the global phasing. It is also at the bin scale. And if I apply the KNN smoothing and the weighted moving average smoothing on the phase the B allele frequency, I can get the panel D here, which is remove some noise. And we apply the XClone in a glioma sample with whole genome sequencing and single-cell RNA sequencing allele information as the ground truth. XClone was able to identify the different allele loss on chromosome 14, matching the reported clones from the whole genome sequencing, while other methods cannot do that. We can show that here on the chromosome 14, we can see the copy loss in one major clone and another allele copy loss in another subclone. But NumBite cannot detect this kind of copy loss. And compared with the ground truth, the single-cell RNA sequencing copy number variation calling results by the XClone are quite accurate and comprehensive. XClone performed good on the copy gain part and the copy loss part, and the loss of heterozygosity part. Here is the summary of the XClone. The highlight of the methods is the three-step phasing strategy in the allelic balance information. And with the phasing in the allele frequency, we can enhance the detection of haplotype-aware copy number variations from the single-cell RNA sequencing data at single-cell level. And in the future, we will inference the tumor clones based on the haplotype-aware copy number variations and may extend to support the multi-omics and may extend to the spatial transcriptomics. Thanks, and looking forward the comments and questions from the audience.